So Dr. Aimee McNamara is an instructor at Massachusetts General Hospital at Harvard Medical School. She completed her Ph.D. in theoretical astrophysics at University of Sydney, Australia. And since then, she's completed a short research fellow position at the Australian Nuclear Science and Technology Organization in detector physics for national security and medical imaging purposes. And after this time, she became interested in medical physics and accepted a postdoc at University of Sydney to transit to the field of medical physics. In 2014, she moved to Boston to work as a postdoc in Professor Harold Paganetti's lab at MGH. And since then, she's been heavily involved in biological modeling and radiobiology. And Aimee, I think first time I met you was around 2011 at AEPM. And then already you were working in radiobiology and if I remember correctly, the role of mitochondria in cell death.
Yeah, that's right. It's
yes.
Around the time I started getting interested. Yeah.
Yes. And Aimee is doing major part of development in TOPAS-nBio. I would say Aimee is behind it.
Yes. Well, yeah, we have a group of postdocs now, so it's a lot more help.
Yes. So please go ahead.
All right. Thank you for the introduction, and also thank you to everyone for having me come and speak today via Zoom. So when I was trying to think of something to present on today, as most of you, I have lots of different topics on the go at once. But I realized that all my projects have a central goal of trying to develop some kind of full mechanistic modeling to predict the response of tissue to radiation. And so that's what I'm going to discuss today is some of those research projects and our work in order to do that. So I thought I'd just start by giving some motivation for why we do this work. So this is in radiotherapy, this is just an ideal situation of what we want to achieve. We want to deliver the most dose to the tumor volume while sparing the surrounding healthy tissue. And so that is the goal of radiotherapy. So this purple curve here is this ideal situation, which is just hypothetical. And then in North America, at least, we have two different types of modalities, radiation modalities, and that's photons and protons. And both of these deliver dose to the surrounding healthy tissue. So you can see that protons do a bit of a better job than photons, where the protons don't deliver any dose behind the tumor volume, but however, they still deliver dose in the. And this can be worse when you look at a realistic case where you have a lot of little proton beamlets to put together a spread out Bragg peak, which conforms to your tumor volume. Then your entrance dose can be quite high. And so my interest is really what happens to the healthy surrounding tissue. What is their response to this radiation rather than what's happening in tumor volume. So there are a lot of medical models that predict. One of the most well-known models is the linear-quadratic model, which goes down to the cell level. So interested in the smallest of the cell, that would be of the tissue. Sorry, that would be the cell. And so the linear-quadratic model makes two assumptions that there are two components to cell killing. The one, the alpha, is proportional to dose, and another beta is proportional to the square of the dose. So then your action is just by the equation here. So alpha is your slope at low doses, and beta is the slope at higher doses. And then we have an imperator, which is this alpha beta ratio, and that's the dose at which the linear components decouple. And this just describes the curviness of the curve.
Aimee, your voice is cutting very much.
An illustration of oh, I'm sorry.
And now you froze the voice.
I wonder, can you hear me now?
Yes, a little bit better.
Oh, okay. That's not good. I'm sorry. Can you hear me?
Yes, now it's clear.
Sorry, it might be my internet. All right, so this is just an example of two different modalities, the survival curves. So this first one is for very densely ionizing radiation. This could be neutrons or alpha particles. And then in comparison, this is a survival curve for photons or X-rays. And these are sparsely ionizing. And you can see that the densely ionizing curve has a much sharper fall off than the sparsely ionizing curve. And so your alpha beta ratio is really characteristic of the type of cell as well as your radiation type. And just to point out that there are many of these mathematical models. This is just one of many. And I would say the linear quadratic model is probably the most well-known. And then because I work in proton therapy, a very important quantity for us is the relative biological effectiveness, or RBE. So that's a concept that relates radiation effectiveness of proton or ion treatments to the very large historic amount of proton data that's available. And so RBE is just given by the dose of photons over the dose of protons to produce the same effect. So RBE is very dependent on this effect that you produce. So this is just to the point. If we go back to our cell survival curves and we look at, for example, an ion curve versus a photon curve, and we want to calculate the RBE for these two modalities, if we pick an endpoint of 0.1 cell survival, we get an RBE of 1.6. But if we pick a different endpoint, in this case, a cell survival of 0.01, we get a different RBE. And so it's important that you quantify this effect as well as your reference radiation, which is the photon radiation. So in the clinic in proton therapy, we use a constant RBE of 1.1, which has been the standard for decades now. But if RBE is indeed variable, does this matter? And so this is just a case study of two different cases, the first being prostate patients, and then the second being a brain patient. And the first two panels, the top panels, show the constant RBE. These are simulated dose profiles. So the dose profiles of RBE is calculated as 1.1. And then the second middle panels are basically a variable RBE. And then the bottom panels just show a subtraction between these two. And you can see that you do get these high dose areas when you consider a variable RBE. And these occur at the edge of your beams. So in the prostate case, the beams are coming in towards the side of the prostate. So you get these higher regions. However, in the prostate region, so the prostate is shown here in the white contour, and the organ of risk here is the rectum, which is shown with the black contour. And these high dose regions do not coincide with your organ at risk. So you would expect no complications in the scenario. However, in the brain patient case, we do find that the high dose region coincides with an organ at risk, in this case, the brainstem. And here you may get some complications, or there may be reason to consider a variable RBE. So the question is, how do we calculate these variable RBE models? So a large proportion of the models are phenomenological models, so they're based on data. And we have developed one ourselves. The proton data set that we used to develop our model was collected by Harold Paganetti and published in a 2004 publication. He went back in the literature and found all proton RBE data that was available, put it all into one single database. And we just did a very simple fit to this database using the linear quadratic model. And we fit RBE to be dependent on dose, the alpha beta ratio, and the linear energy transfer, which was dose averaged. And this was the equation we came up with, where these p-values are just simple fit parameters. And we found that the RBE is a function of dose, it decreases. RBE is a function of alpha beta ratio, decreases, and also it increases linear as a function of LET. So this is pretty much in line with what the other models find. So to be honest, there was nothing surprising there. We just had a more complete data set. So when we compare our RBE model to some of the other phenomenological models, our data set just contains the data that they used as well. So it's all subsets of the same data. Our one just includes all the data. So this plot here is a simulated SOBP profile in water. The black curve is the physical dose with no scaling factor. This red curve here, if you can tell it's red, it looks a little orange, is the RBE, which is scaled by 1.1. And then all these dashed lines are just the different RBE models. And you can see they all make very similar prediction, just varying levels of what the actual, the increased dose is. So we find that we have this increased dose at this distal fall off. And the reason for this is because of the LET. These models are all dependent on LET. And this is just a overplot of the LET on top of the SOBP. You can see at the distal edge, the LET is quite high, which means that you get this high dose region. And so in the clinic, this is maybe only significant in the case where you have an organ at risk, which is directly adjacent to your tumor volume. And that would be the only time you would need to really be concerned about that. The other limitation with phenomenological proton RBE models is the data itself. So this is just to summarize the data in that Paganetti data set. The vast majority of the data is from non-human cell lines. And the human cell lines all happen to be cancer cell lines. And there is just this very, very small subset of the data, which is healthy human tissue. And so that is the tissue that we're most interested in because we're concerned about the organ at risk rather than the tumor volume itself. And the majority of this data is also in vitro data. There is some in vivo data, but it's a very, very small subset. And we, of course, know that the in vivo situation is much more complex than in vitro. And so there is a need for better data for proton RBE. And then just to also point out that there are other models which are not phenomenological. One of these that applies to proton therapy as well is the local effect model. And these are based on assumptions of your local dose. And the local effect model assumes that local doses deposited within the same volume should lead to an equal effect independent of the radiation quality. And so the local dose is derived from track structures where the actual energy deposition events are determined from the track itself. And in these models, the cell nucleus is usually determined to be the sensitive target. I'll also point out that the newer versions of the local effect model go even further to look at DNA damage and double strand breaks. So this is just very much summarized. But it's based on the local doses. So if you look at the carbon ion dose profiles, you can see that carbon ions have a very densely ionizing track. And this would lead to a higher RBE than if you compare this to the photon case, which is more homogeneous. And the local effect model is mostly used for ion therapy, and it's actually used in treatment planning in Germany. And so if I was to summarize radiobiological approaches that we currently have, and then overlay it with what we ideally would like to have at the bottom here. So this would be our full mechanistic modeling scenario, where we would be able to not only model the track structure of our particles in high detail, including not just physics processes, but also the chemistry processes, to predict biological damage, such as double strand breaks. And then this biological damage could then be used to predict a biological response within the cell. For example, the onset of DNA repair or signaling by the cell. to predict some kind of cell response. Usually this is apoptosis. And then the missing piece is to somehow go from the cell response to what the tissue response is, or toxicity. So if we look at what models we have, none of them accomplish this last step of going from some kind of cell survival response all the way up until the tissue level toxicity. And so the LQ-based models really only cover the cell response. The local effect model, which I detailed, does a better job. It takes into account the track structure of your particles all the way to predicting your cell response. And then just to point out that there are some other models that I haven't mentioned and won't go into detail that also accomplish different levels of this whole full mechanistic approach. All these models are mostly used in ion therapy at the moment. So what would the mechanistic approach look like? So in the case of proton therapy, again, we would start off, again, maybe focusing on DNA, although it shouldn't be limited to nuclear DNA, in my opinion. We would use track structure simulations to predict the distribution of damage in the DNA. So in theory, proton therapy will create denser tracks with more clustered DNA damage, so clustered double strand breaks compared to photons, which are more sparsely ionizing. This could in turn initiate different repair pathways, which would then lead to different chromosome aberrations, for example, and potentially different survival, but also different tissue effects, such as signaling effects, inflammation, also bringing in more complex systems such as the immune system as well, and the circulatory system, and then somehow linking this to what the organ response would be. And so the idea would be, by understanding the differences on this track structure level, we could predict different outcomes in different organs, which is sometimes what we see in the clinic, where we have different types of toxicities between protons and photons, or different levels of toxicity. All right, so that's ideally what we would like to have. We're not quite there yet, but we do quite a good job of some of those steps already. And so one of the things that we can do currently is the track structure modeling. So we use a Monte Carlo simulation approach for this. The difference between standard Monte Carlo or condensed history Monte Carlo and track structure Monte Carlo is illustrated in this figure here. So if you had to do a standard simulation using just your standard GM4 toolkit of an electron track, it would look something like this. So this is in just a very tiny volume, a couple of nanometers in size. But if you looked at the track structure models, you get a much detailed description of your electron track. So these two scenarios produce the exact same amount of energy deposition within your volume. So there's no difference in that. They have the same numbers. But the GM4 DNA processes give you a much better idea of where those energy depositions take place. And that's really important when you're looking at very tiny molecules such as DNA, which is a couple of nanometers in size. And you want to predict something such as double strand breaks. And so the GM4 DNA processes is what we use in our simulations. And they do a step-by-step interaction of particles. And this is mostly in liquid water, all the way down to your EV scale. And also just to point out that the chemistry is also very important when you go down to the scale. And when you start to look at heavy ion therapy, this can become very important because you have very complex tracks. And so we developed a toolkit, which is called TOPAS-nBio. It is a wrapper for the GM4 DNA toolkit, and it is an extension to our other existing toolkit, which is known as TOPAS, which is being used extensively in medical physics applications. And the whole idea behind TOPAS-nBio and TOPAS is that we make it easier to use for users that may not have advanced programming skills. So anyone who's familiar with GM4 knows that you have to have quite advanced C++ programming skills in order to use it. And we wanted to take away that restriction, especially in medical physics, and also then for radiobiology, to attract radiation biology researchers to be able to use the toolkit. And both of our toolkits are freely available. TOPAS-nBio is completely open source, and TOPAS will very, very soon be open source. At the moment, it's not. And both are extensively documented. So we have a read the docs for both our toolkits. So I guess the selling point for TOPAS is our parameter system. So
all right, books.
Exact same parameter system. It's based on text files. And the whole idea is to change parameters, not code. And so your parameter files will specify everything from your geometry, your particle sources, your physics, and all of this has been tested extensively by us for medical physics applications. So we undergo a rigorous testing procedure where we will do our regression testing specifically for medical physics applications. And so this is just an example of some of the applications you may use TOPAS for, sorry. For example, you could maybe use it for patient QA, where you replace a measurement with a Monte Carlo measurement. It has been used a lot in proton MRI studies, where we look at the feasibility of using MRI, where Monte Carlo can predict your beam deflection caused by your magnetic fields. And it's also used by us extensively for range analysis calculations, comparing our Monte Carlo simulations to our treatment planning system. This can help you understand any uncertainties in your treatment planning, especially when you have heterogeneous tissue, for example, in nasal cavities or lung, which may require larger margins of error. So what would be the difference between TOPAS and TOPAS-nBio applications? Well, TOPAS-nBio applications is really looking at the biology. It's not clinically based at all at the moment. You would be looking at simulating DNA damage to your nuclear DNA or just simple plasmids, DNA strands, for example, if you're running biology experiments and you want to understand distributions of damage to those molecules. We have a lot of users that is currently using it for nanoparticle simulations, where they want to try and understand what the dose distributions look like around the gold nanoparticle. In relation to some biological molecules, again, DNA. But we also have users that are using the toolkit to look at damage to other biological molecules, such as membranes. We also have a few users that are looking at neuron simulations to look at damage to neuron synapses. So when we look at what we need to model when we're looking at radiation damage in cells, the first is the physics processes, which occur almost instantaneously upon irradiation. And this is damage directly to your molecule, for example, if our target is DNA. Then the other thing we need to be concerned about is chemistry reactions, which occur at a slightly later time point. And the most significant chemistry processes that we consider are radiolysis of liquid water within the cell. So you would have the radiation interacting with the water molecules, which would create some free radical species. These free radical species can then diffuse throughout the cell and interact with your target volume, causing damage as well. But then, of course, this is just the types of damage that you can calculate. Then, of course, there is damage to other molecules as well within the cell. So looking at different organelles, for example, the mitochondria could be another target. So this is basically an overview of what TOPAS-nBio can do. So starting with the physics stage, it uses the GM4 physical models to look at any modeling ionization from incoming and secondary radiations within your biological medium. The physics stage can then feed into the chemistry stage, where you have some radical species production and the diffusion of these species. All of this can be done within geometries. And so we offer quite a big library of different biological geometries in which you can run your simulations, which I'll go into more detail a little bit later. And then through the physics stage, you can predict the direct DNA damage, for example, and also indirect damage from your chemistry within your geometries themselves. And then we offer cell repair modeling currently. That's our biological stage. And this includes different DNA repair models that you can use. And then from there, you can calculate micronuclei formation, foci formation, etc. And then something that we don't do currently, but it is our end goal, is to have some endpoint calculation where we connect this stage to some kind of outcome modeling inside your organ or your tissue level. So just a little bit more on the GM4 DNA processes. So TOPAS-nBio is completely based on GM4 DNA, which is already a large collaboration. There is a lot of ongoing work within the GM4 DNA collaboration. We work very closely with the collaboration, and we feed back a lot of any of our developments back to them, particularly in the chemistry. So Jose is within our collaboration, but also is now the head of chemistry in GM4 DNA. So if you're interested in any of these models, they have a lot of publications on the DNA models, but also on the experimental data that they use to validate these models on or base these models on. So ongoing developments within the collaboration is there is continuous work on the physics processes. These are very hard models to develop because they are going down to such very, very low energies. Currently, all of the experimental data is in liquid water, although there is some data now coming through within DNA materials, which is what we need. We don't need liquid water, we need the realistic materials. The chemistry processes is an ongoing area of development, which I'll also go into a little bit more detail about, but also improving the molecular geometries for DNA. DNA is very complex, and how we package it within the cell and how we model it is important for predicting accurate distributions of damage. So a little on the chemistry, just want to point out, I'm not an expert in the chemistry at all, so Jose has done all this work within our group. But the most important chemistry models from a biological standpoint is any interactions of radiation with H2O, which causes the formation of these free radical species, which are very damaging to biological molecules, particularly these OH ions, which cause significant DNA damage. So we focus on these in most of our simulations, although the simulation does produce a very large range of different types of free radical species. So you could concentrate on different free radical species, for example, perioxide, if you're interested in membrane damage, for example. And these chemical reactions can be quite complex. So this is just a summary of all the different reactions that we include in TOPAS-nBio. At the time that this publication came out, a lot of these processes were not available in GM4 DNA, but we have fed them back to the GM4 DNA collaboration. So I believe they're all available in both toolkits. And then Jose has done a lot of validation work in order to get these numbers to be as, these reaction rate constants to be as accurate as possible, which is also extremely challenging because the experimental data is quite limited. And we collaborate with radiation chemists where we can, particularly Jay Laverne at the University of Notre Dame, which he provides a lot of feedback on these models. Also, Jose has managed to get the chemistry to work within our geometric models, which is important when you want to consider damage to different types of DNA, for example. So this is just a little video that illustrates a simulation of a proton in a liquid water medium. So the first stage that you see there is the physics stage, and then the second stage is the chemistry stage. So I'll just let it rerun. So what you see is the proton coming through in blue, secondary electrons coming off the proton, and the yellow dots are actually ionization events. And then a later time point, the chemistry kicks in. We have different probabilities of different free radical species forming. They are just color-coded according to this little plot on the bottom, the legend. And then they diffuse through the medium and interact with our geometry, which is just a simple plasmid ring, DNA plasmid ring. And the diffusion is just based on simple Brownian motion. And then a little bit on designing the geometries, which I was heavily involved in. So something that I wanted to achieve within the toolkit was that we didn't just focus on DNA specifically, but we would allow users to go from a macro scale simulation, so that's on the cell level or even the nucleus level, all the way down to your DNA strand level, your double helix strand. So we, of course, have focused on DNA because that is a very important target for radiation. But we also are interested in non-nuclear targets, so different organelles in the cell, because those could be just as important as the DNA. And so DNA is packaged within the cell using a very complex folding. And we need to capture this whole packaging in order to accurately predict DNA damage. So just going back to the cellular organelle level, so we have several different cell models. This cell model here is just a simple spherical cell for illustration. It's what I call the physicist viewpoint of biology. But we wanted to move away from just using very simple spherical cells. They're very useful, but they're not realistic. And so we have some realistic geometries which are based on microscopic images of cells that are very commonly used in radiation therapy. This is an example of a fibroblast cell, which usually in vitro, they're very flat. They have all these little arms that come off the cells, and they can move around quite significantly within your petri dish. So that's a more realistic viewpoint. And then we also offer geometries for specialized cells, for example, neurons. This is just an illustration of a neuron within TOPAS-nBio. You also have control over your geometry within the parameter phase space. So you can position, for example, your nucleus wherever you want. You can add organelles to your cell. You can have different numbers of organelles and also different positions. These are all just randomly arranged, but they don't have to be. We also offer the option of randomly placing our objects within, for example, a box in this case. So you have cells within a box so that you can simulate any kinds of cell experiments, for example. And then it was very important for us to have more accurate models of the nucleus, because this is a very important consideration when you're modeling damage to DNA. And so it was important to capture the nucleus details, not only on the nanometer scale, but also on the micrometer scale. And so generally cells in S phase, you can see they have these chromosome territories. This is just an example of a chicken fibroblast showing the different chromosome territories. And these occur at specific regions within your nucleus, and also the chromosome territories are in specific positions relative to one another. So this can be really important when you're trying to predict damage to particular genes after irradiation. And in 2009, this paper came out in Science that showed that if you have a very long string, which is presumably your DNA, and you just fold it randomly, you don't get any kind of structure in your ball of string, if you like. But if you fold it using a fractal folding scheme, you do get the structure, and it's very similar to what you see in real life, which is these chromosome territories which occur at different regions within the cell. And so we set out to mathematically reproduce that within our models. And so we developed a nucleus which is just based on a 3D Hilbert space filling curve. So this little video here just shows what a Hilbert space filling curve in 2 dimensions, where you have a square, and with each iteration you fill the space using the repeating shape. And then you can do this in 3 dimensions, where you use an open cube, and with each iteration you can fill the space. And this also fills the space very efficiently, using as much of the space up as possible. And so we use this to fill our spheres. So this is just a very simple illustration of that. And so we have our fractal model within TOPAS-nBio, which looks like this. And this one has been color-coded to show the realistic chromosome territories, which have been just simply numbered and colored. So I want to point out that each of these little cylinders that compose this, this big blob, if you like, also contains a full chromatin fiber geometry. So this also has your histones with your double helix, sugar phosphate backbone and base pairs wrapped around it. And there are several models for our chromatin fibers available within TOPAS-nBio as well, depending on your applications. And then from here, you can simulate different types of damage, from direct damage, but also indirect damage from the chemistry. And then we also have other models that are available, like I mentioned. So that was our fractal walk model, but we also have some simpler models. This is just a model based on where the chromosome territories are just simple cubes placed within this ellipsoid. And then each of these ellipsoids, each of these cubes contain these little flower shapes, which represent our chromatin loops. And each chromatin loop then contains our double helix structure as well. So this is a simpler model. There are benefits to using these, mostly in the processing time of your simulations. And so we have this extensive library of different geometries that are available. This is just showing some of them, where we have our cell models, our nucleus models. We have several nucleus models that are under development because these simulations are very time consuming. They consume a lot of memory. So we're working on improving that. That's an active area of development. We also have these organelle models, membrane models, which are important for some studies as well. And then we also link to a few different databases. So this is just an illustration of an atomistic viewpoint of a DNA segment, which has been taken from the protein database. We also link to a neuron database so that you can pull different geometries from the neuron database and input them into TOPAS-nBio. So those 2 databases allow you to simulate multitudes of neuron geometries or proton geometries, depending on your application. We've also done some work in looking at validating our models. So mostly what we've done is compare TOPAS-nBio to other track structure simulation codes. It's hard to find experimental data. So it was our first starting point, was to make sure that we agreed with the other codes that were available. Really, we're comparing GM4 DNA to these other codes rather than TOPAS-nBio. Many of them share the same cross sections, which is mostly data for liquid water. However, some are based on cross sections for gaseous water. So we do expect not as good agreement with those data sets. But we do find that the CPA code and GM4 DNA agree the best, which use the same cross sections. CPA 100 is quite an old code and it hasn't been updated for many years. So there's some differences there with improvements in the modeling. So even though experimental data is very difficult to come by, we did make an attempt to try and find data. So one of the challenges is separating the chemistry from the physics when you do these validation studies. So ideally, you would need to do irradiation of DNA in a vacuum, which is very challenging to do. So the closest thing we could find was some plasmid irradiation experiments in dry plasmids. So in theory, no liquid water, but there could still be water in the air, for example, that could affect your results. And we do find that our single and double strand break simulations agree show similar trends to the experimental data. Although these numbers were slightly off, it's within the experimental error. And then I mentioned that we can also do DNA repair modeling. So we link back to a few different models. Most of the DNA repair models are analytical models, and they focus on a single pathway or endpoint. This is a very active area of model development, and we work very closely with Stephen McMahon, who's at the University of Queen's in Belfast, and also the University of Manchester group, which have been doing a lot of model developments for DNA repair models within Geom4 itself. And so they have contributed their model to TOPAS-nBio, so you can model DNA damage and repair now within a single simulation using their code. Stephen McMahon's model is a separate model which you can plug data from TOPAS-nBio into it. It's Python based and it's quite simple to use. But we are doing a lot more developmental work on this. So this is just a study which was done by one of our PhD students, where we put all of this together, if you like, where we look at doing DNA damage and repair modeling in a single simulation. In this case, we only looked at Stephen McMahon's model for DNA repair. And so this here is just a comparison of single strand break to double strand break ratios for TOPAS-nBio to other Monte Carlo simulation codes for our full fractal Walker nucleus model. And then we also looked at residual fraction as a function of time, and misrepair or residual fraction as a function of LET as well. So this is all for proton simulations. Again, just using a single model. And then finally, using those predictions, we compared back to experimental data where we actually calculate realistic measurements such as dicentric fragment yield and also acentric fragment yield per cell. So some of these experiments, as you can see by the dates, are really, really old experiments, but again, the experimental space is very limited. So our simulations are shown here by the upside down triangles and the squares in both plots. And we find better agreements if we only take double strand breaks, which are greater than 3 megabase pairs. And the reason for this is the experimental data was taken by eye. And so we can assume that double strand breaks less than 3 megabase pairs in distance may not be observable. So we do find some good agreements, and we definitely see similar trends to the experimental work. There is still work to do in this space to improve the models for sure, though. I also wanted to point out that when we were doing a lot of this work, we found that it was very difficult to compare to experimental data, but also to share data between different groups that are also doing very similar simulations. And so we proposed in 2019 a new standard to share DNA data, just to make collaboration so much more easy. If everyone's data is in a similar format, we can use DNA repair models more easy between different groups. And so this was the standard that was proposed, and it's a big paper where we invited almost everyone that we knew working on these types of simulations to get involved. And it turned out to be quite a complex data format. But if anyone's interested in it, there is this publication on it. And then just to point out, we have been comparing our simulations to other groups that do very similar simulations to us. And we did a comparison. So this is still under submission, but we do find quite large differences between the different groups and probability of double strand breaks within our track core versus the double strand density along tracks. This was all for proton data. So we started the study just looking at protons. And the point of this whole study was to show that the assumptions that you make in your models, even though they seem to be very small, can greatly affect your outcome data and can differ between different groups quite significantly. So yes, there's a lot of work to still be done. We need more experimental data to really understand what parameters we should be using and what assumptions we should be using in our models. And then I thought just lastly, I would come back to how we can apply this to something more realistic, so something from the clinic. So one of the projects that I've been working on is looking at rib fractures in the clinic. So we found that our proton patients experience a higher probability of rib fractures than our photon patients. So the cohort we were interested in particular was breast cancer patients, which saw about a 7% chance of a rib fracture compared to the photon cohort, which only saw about a 1% chance of fracture. And so we set out to try and understand this a bit more. So we did just normal Monte Carlo track structure simulations on the patients that had received rib fractures by contouring the rib fracture and looking at the dose and also the LET weighted dose within the fracture region. And so the LET weighted dose here was just used as a surrogate for an RBE. And we find that when we use an LET weighted dose distribution, we do find a higher region, a higher dose region, which coincides with the fractured rib. So one reason for these higher fractured ribs may be because of that distal edge of the fallout region coinciding with the chest wall of the patient. And this is just how we do our treatments at MGH. Other centers that don't treat with the same beam alignment, where the distal edge coincides with the chest wall, do not see these high incident rates of fractures. And so this led us to question why we might see a difference in fractures between protons and photons going back down to the cellular level. And it turns out that in bone, there are these very specialized different bone cells, osteocytes, osteoblasts, and osteoclasts, which have very unique geometries, which make them a perfect candidate for Monte Carlo studies. And so in particular, osteoclasts are these giant cells which have multiple nuclei, which in theory, if you damage one or two of the nucleuses, other nuclei can take over and function for the cell. Osteoblasts are very small cuboidal cells which occur only on the outside of the cell cartilage, and osteocytes occur within the bone matrix. So just looking at just the cell geometries, nothing else, we find that osteocytes are much more likely to be damaged than osteoblasts in a very simple bone matrix setup. And so we also set out to test this doing some experimental data. And so this was just some preliminary results looking at survival fraction of osteocytes, which we focused on because of our simulations, looking at difference between X-ray and proton irradiation. And we do indeed see that protons cause a lower survival fraction within these cell types. But we also see that the damage seems to persist longer in proton irradiated cells than in X-ray irradiated cells. So this was just looking at foci formation. So this is an example of 53 BPI foci within our osteocytes. You can see the different foci which coincide with the DAPI stain of the nucleus. So you can see that these do indeed occur within the cell nucleus of the cells. And these seem to, the proton cells seem to have a larger recovery than the X-rays. So I just wanted to conclude by basically saying that we have a lot of work to still do in this space. So we focus on RBE because it's a concept which relates radiation effectiveness of proton and ion treatments back to that large amount of historical photon data. So that's why we always go back to an RBE concept. But RBE is definitely flawed. It's mostly determined from colony formation, foci formation, micronuclei formation, which are all in vitro studies. very few in vivo studies, which would probably form a more complete picture of what's going on within our tissue. And of course, in the clinic, the relevant endpoints are not any of these cellular endpoints such as foci formation, but it really is some kind of tumor control or normal tissue complication. And at the moment, we have no way to link these two scales. So we have a huge scale mismatch in our modeling. So just to summarize, I do think that we do have a full mechanistic approach is possible, but it does not exist yet for a tissue response. It does exist for a cell response, but we need to get somehow back to the tissue response. And our modeling approaches start with track structure simulations, different biological target models, which we need to keep improving. We also need different targets from just the DNA, because of course we know that other parts of the cell are also important in inflammation response or cell signaling or bystander effect as well. And we also need to model those other systems, such as inflammation, which is important in toxicity in normal tissue. And then just want to also end by acknowledging all the other people who have contributed to this. So the rest of the TOPAS-nBio team at MGH, but also UCSF, and also Joseph Pearl at SLAC. We work very closely with the biologists at MGH, namely Kathy Held, and also some of the postdocs who are taking some of these experimental data for me. So Yunkako is involved in the bone project that I'm working on. And then also we get a lot of feedback from, luckily from our clinical team, who are also interested in answering these questions with us, which is rare. And yeah, I'll take any questions if anyone has any.